Thank you for your introduction and thank you for the invitation of Professor Sankava. Today I would like to talk about the extraction, the quantitative information from the temperature-dependent near-infrared spectra. The research interest of our group is chemometrics. We developed several methods for processing the analytical signals, such as yesterday I mentioned the wavelet transform. From 20 years ago, we moved our research to the analysis of the near-infrared spectra. We developed a lot of methods for modeling, for calibration, for determination the calibration samples, and several methods for pre-treatment of the signal. Particularly attention is the variable selection is a very difficult problem. We developed several methods. And recently we did some work about the model transfer. It's related with the application of the spectroscopy. We did also some applications in industry and the quality control. From 10 years ago, we did a project about the microanalysis using the near-infrared spectroscopy. We used the adsorption as the pre-concentration technique to increase the sensitivity or to improve the detection limit of the near-infrared spectroscopy. This is a defect of the technique. In these studies, it was found the changes induced by the perturbations, induced by the interactions in the analyzing system can be detected by the spectra. So we consider that if we take every perturbation as a factor to affecting the spectra, it may be modeled. We can use the change caused by the interaction as the source of the signal to do the quantitative analysis. So we proposed the temperature-dependent near-infrared spectroscopy. I think this is just the idea of the Aquaphotomics. It is a similar idea. So we started such work from maybe 10 years ago, maybe 8 or 9 years ago. Recently, we started another two work. One is the development of the small instrument based on the chemometrics algorithms. And we also started to considering the method for the big data system. Today, I would like to talk about the temperature-dependent near-infrared spectroscopy. The temperature effect of the spectroscopy is an old problem. A lot of work have been done for the problem. But it has been, I think the defect is a defect. A lot of work has been made to remove or to correct the temperature effect. But if we think the reason, if we think the cause of the effect, the effect is caused by the change of the structure or interactions in the system. And such changes is related to the composition and the property of the analyzing system. So the quantitative relationship should be built between the spectra and temperature, concentration, and other properties. In fact, a lot of techniques have been developed besides the temperature-dependent near-infrared spectroscopy. The AIS spectroscopy and other spectroscopy techniques have been developed. Such techniques we can name it as the perturbation spectroscopy. The application of the temperature-dependent spectroscopy has been studied 20 years ago. The Professor Ozaki and Charnetsky did a lot of works by using the chemometrics method to the structure analysis, particularly for the hydrogen bonding analysis about the alcohols, waters, and proteins, etc. Professor Brew has used the perfect is a three-dimensional chemometric method for the quantitative analysis. But the aim of this paper is also to correct the temperature effect. They build the quantitative models for the using the spectra measured at a different temperature. Maybe 2005 or 6, from Professor Zankova proposed the concept of Aquaphotomics. She takes all the changes in the system as a perturbation and measure the spectra under these perturbations, and then to explore the information from the spectra. I think this is the idea of Aquaphotomics. There have been studied a lot of systems about the equations and proteins, milk, blood, and urine. Our work starts from very basic things. At the beginning, we studied the temperature effect of these three groups. This is the basic response for the spectroscopy, for the near-infrared spectroscopy. This is the spectra of water. If we measure in different temperature, a regular change can be found. And also for the methanol and the NH group, and even for the CH group. If we enlarge the spectra, a regular change can be found in the spectra. This work was mentioned by Professor Koch yesterday. We built a lot of models for different systems, including the water and the mixtures. And this model is very good. It's interesting. The R can be as high as the 0.999. So the more interesting thing is if we use this model is built by the spectra of water to predict the spectra of solutions. This is the solutions containing different concentration of ethanol. We can also obtain the straight lines. But if we compare the lines, the intercept and the slope of these lines is different. If you compare these intercepts and slopes, another straight line can be obtained. This is the relationship of the concentration and the intercept. So this result gives us confidence. We can obtain quantitative information from these spectra. So in our recent works, we concentrated our effort in developing the chemometric method for obtaining the quantitative information and the structural information. Generally, we have the 2D to two-dimensional data matrix. We have different sample and measure at the different wave number. But if the temperature is involved in the experiment, we have a 3D array of the data. And if the other factors involved in the experiment, we have higher dimensional data arrays. So we must develop the high-dimensional method to extract the information from these data. Up to now, we have tested the several methods. All these are high-dimensional methods. And we need more work to explore the new method. The first method we tried is the multi-level simultaneous component analysis, MSCA. This is the model of a two-level MSCA model. This model can describe the data measured under the two affecting factors. For example, each X block is measured under the different temperature, and each block means the different sample. And then the first level model, from the first level model, we can describe the changes caused by temperature. And the second level model, we can describe the changes caused by the concentration. So we can get both the information of temperature and concentration. So we did an experiment. We prepared 5 different solvents, and we prepared 9 samples containing an analyte. And we measure each sample at the temperature from 30 to 60 degree at a step 5 degree. In this way, we can obtain the 5 3D data. For each solvent, we have a 3D data matrix. In the calculation, we can take the data measured on 7 temperature as a block, as a X block, and we have 9 blocks. So the first level describes the change of temperature, and the second level describes the change of concentration. This is the result. For each solvent, we can obtain a straight line about the temperature and the scores of PCA. So this means the spectra contains the quantitative information of temperature. So we can predict temperature using this line. And if we compare the slope of these 5 straight lines, we can obtain this line. Now this means the composition of the solvent can be predicted. This is the result of the first level model. And if we examine the result of the second level, we can also get the 5 straight lines. But each point includes the 7 temperatures. This means in the second level model, the temperature effect was completely removed. The temperature, all the 7 temperatures get the same point. So the temperature is completely removed. And similarly, if we compare the slope of the 5 straight line, we can also get this one. So this is also means the composition of the solvent can be predicted. Following this study, we used the method for quantitative analysis of glucose serum. We prepared several. The one is measured with the prepared solution of the glucose, and the data set 2 is prepared using the serum sample. These samples are collected from the hospital, and data set 3 is also obtained from the samples serum, but it's collected in different time. And the spectra was measured also in this temperature range. And we get a 3D data matrix. The one dimension is concentration. One dimension is the wavelength of the wave number. And the third dimension is the temperature. This is the spectra. This is the raw spectra measured by the instrument, and this is the second order derivative of the spectra. This spectral range is the absorption of water, and this region is about the proteins. Is about proteins. If you compare these spectra, we can see the information of the protein, and the most interested region is the spectra of water. So I just tell you the result obtained by this region. This is the first level spectra, first level model. Now we can obtain the loading and the scores. From the loading, we can see some of the structural information. But in the score, we can get a very good straight line for the temperature and the coefficient. And in the second level model, we can get 3 loading and 3 scores. And in the second PC, we can obtain this straight line, which can be used to predict the concentration of the glucose in serum samples. This is the glucose solutions. You may ask why not the first PC. This is the interference from the water vapor. This experiment was did in the summer, so we didn't use the nitrogen gas to flow the gas. So this is interference. So the second PC is the main information of the water. This is the result for the serum samples. It can be seen in the third PC, we obtain this straight line. And all the first PC and second PC is the interference from the water vapor. This means the spectral information of the glucose is very small. It's even compared with the interference from the water vapor. So it's smaller than this signal. So it lies in the third PCs. This method is proposed in this year. We arrange the data blocks in this way. Each block either is a sample of a concentration with measured at different temperature, or all these samples measured at a temperature of different concentration. If we align the data blocks like this, if we do the PCA to this data matrix, we can obtain the scores and the loadings. All the samples share the same score, and all the difference is contained in the loadings. So we can tell the difference between the samples by the P. If we take one of the sample as a standard sample and calculate the standard signal by this equation, and then we can use this equation to calculate the comparative the difference between samples by a parameter. This parameter is a quantitative description of the difference. So this is our experiment. Glucose solution with different concentration, and there is a 22 serum samples from the hospital and measure the spectra at different temperature, and we get 3D data matrix. And we can calculate in two ways. One is align the data matrix along the temperature, or we can align the data blocks according to the concentration. And this is the result. We can see the parameter shows the difference of the temperature. The spectral variance caused by the temperature is a linear relationship. And in this figure, we can see the concentration with the spectra. It is also a straight line. And these two figures are obtained from the prepared solution. And this figure is obtained from the serum samples. We can see the straight line can be obtained, not so perfect as this one, but it is accepted. It can be used to predict the concentration. Last year, we also did a comparison about the high-dimensional decomposition method. These are a traditional method proposed by the other groups. We just test the application of this method for processing the temperature-dependent spectral data. Theoretically, such methods should give us more information. They can give us the structural information from the loading, and it can give us the quantitative information in the two scores. One is the temperature and one is the concentration. But in the this is experiment is we prepared two data sets. One is the 3D data set, a different concentration sample measured at a different temperature, and this is the I mentioned before we prepared the samples with the different solvent, so we have a 4D data matrix. The result is not so good, I think. We can find some structural information about the water species, but it is difficult to find the information of other water species. So this is the information of quantitative result. This is good. And even if we use the multiple linear regression, we can get a very good quantitative information from the spectra. This is the result for the 4-dimensional data. I have no time to introduce the. We have a poster. Maybe we can discuss in the poster section. I think it's more different to do the structural analysis compared with the quantitative analysis, because we concern more about the structure change in different systems. But recently we did some simulation. It is very, very complicated about this peak. So we have also a poster about this subject. This is a result in our work for the resolution enhancement. We used the wavelet to calculate the high order derivative. We can see the structure, the detailed information of the water. And we also developed several methods for separation of the component spectra from the mixture. We find at least 6 peaks from the water. This peak changes with temperature. And this is a new method for selecting the temperature-dependent variable in the spectra. The result is very interesting. For different solutions, we can obtain different results for the variables sensitive to the temperature. So this paper has just published in the ChemLab. So you can see the detail from the spectra, from the paper. So finally, I think the near-infrared spectra provide us a very powerful tool to measure the spectra of the water system. But the spectra is generally like this. So it's difficult to see the details contained in this spectrum. Aquaphotomics is a new discipline which understands the problems from a new perspective. So it's really interesting. It's a new direction. We must do a lot of work in the field. But I think the chemometrics method is needed. We must develop chemometric method to explore the information contained in the simple spectrum. So a lot of work is waiting us to do. So I think we must work hard for the field. So I just thank you.
So thanks, Professor Shao, for your interesting talk. We have time for at least a couple of questions. Does anyone have a question? Juliana?
Thank you very much. You gave few new directions, and I really, I think what you showed about humidity might be very interesting. And one perturbation that you have tried until now is temperature.
Yes.
Do you think that if you change humidity in a way you change temperature, you might be able to obtain better results in terms of accuracy of single components, and also you might be able to unravel new water absorbent bands?
Mm-hmm. Yes, but it's difficult to control the moisture in the experiment. So if we have equipment to control the water content in the air, it could be.
Yeah, but I mean, you can. I know that it's very. If you can at least measure it, you need a change.
Yeah.
And you need a measurement, because you activate the system through the humidity, and then when you analyze the results, then you will see the activated water bands. I think.
Yeah. Yes.
Thank you very much, really.
Okay, you have a question, please.
Thank you, Professor Shao. So at the beginning of the presentation, there's one slide you talk about 4 dimension, and there's one variable you list, pH. I assume that's intentional, but you didn't talk about it much afterward.
Yeah, yes.
I wonder why you think pH can be, like, what's your thought process behind you think pH is possible to be detected? Thank you.
Yes, that's the work we are doing. So we have not get any results from we are still doing the experiment. The pH is a factor to affect the structures in the solution. So if we change the pH gradually and measure the spectra at different pH, I think it's the idea to get the structure change with the pH.
But does that imply the pH can be chemical independent? I mean, you use different type of chemical to manipulate the pH. You can still extract the information just about the pH, but not the chemical compound you use to change the water's pH.
No, the aim is to find the structure at different pH, because the spectra, if the spectra measured at different pH, we can get the structure at each pH, and also the structure change with the pH.
Okay, thank you.
Well, I have a quick question myself, or more rather a quick comment about it's myself. about the MSCA. So actually, it's more like an advice. So the MSCA is a method is a method for so-called repeated measurements design. So it takes advantage of the fact that you are measuring the same sample over a factor, so that the same sample be the contour of itself. So actually, if you would revert the treatment, so if you put temperature as a within and sample as the between, then you would probably have a greater advantage in the configuration of the experimental design, because this is the way it should be done.
Yes, in our paper, both the alignment was reported. Here I just showed one of these results.
Okay. Well, thanks once again very much for your talk. So, and the next speaker is Professor Yasuo Mitsukura from the Department of